discussion of the osmosis, and people go in terms of the cell structure sometimes from inside to outside, from outside to inside. I prefer to go from the inside to the outside portion. All right. So the inside structures. Some of them cell cannot exist without, like nucleoid, which you can see right here. It's circled with a green color. It's accumulation of the DNA, cellular DNA that forms chromosome. In prokaryotic cells, in the bacterial cells, we usually have one chromosome. And DNA in the chromosome is actually wrapped around the nucleoid associated proteins, which means that when it is wrapped around the proteins, quite literally, it is silent. In order for the gene to get expressed, that fragment of the DNA has to unwrap back. Make sense? Well, cytoplasm is another obvious structure that cell cannot exist without. This is your cytoplasm. Okay, something that fills up the cell. You have no cytoplasm, you have no cell. It's functionally, you can say it's a structure, it's a storage, it's the place for everything else. Okay? Ribosomes. You can see ribosomes illustrated here. Every bacterial ribosome consists of two units. The larger 50S and the smaller 30S subunits. When they are put together, they form 70S ribosomes. Those units, the S units, the Swedbergs, they are not additive. So 30 plus 50 in this case is not 80, it's 70. These units represent how fast a particular structure, in this case subunits of the ribosome, can precipitate. So like 70S is big and it will fall down faster than 30S. That makes sense? Functionally, of course, ribosome is protein translation. Sorry, RNA translation. Protein synthesis machine. Ribosomes in eukaryotic cells are very, very different. And if they're structurally different, what can you tell about the way they work, the function? Yes, they have a lot of differences. Of course, the final outcome is the protein anyway, but they have a lot of differences. Now, if they are functionally slightly different and structurally slightly different, what can we do with bacterial chromosome without affecting eukaryotic chromosome? Well, we can inhibit it. Yes, we can do something with the bacterial chromosome to stop it from working. Does that make sense? That will not affect the eukaryotic chromosome, which means we can create drugs that specifically inhibit translation in bacteria, but not in humans. And a good portion of antibiotics work this way. They attack, they target the bacterial chromosome. Inclusions. Storage. You can see different types of inclusions. You can find organic molecules there. Storage of food. Storage of sulfur sometimes as a source of electrons in oxidative reactions. Here you can see magnetosomes. Inclusions that contain iron. Magnetotactic bacteria that follow the magnetic field of Earth have those inclusions. Why do they do that? We have no clue. Endospores. Now this is the page that you do not have. It's on the blackboard. You can print only this one page. Additionally, it's only one page that I completely forgot about. Okay, it was updated. We're gonna talk about endospores, and I'll give you the quiz. Endospore has nothing to do with the spores of fungi. It's a completely different, separate structure. So it allows microbes to survive, okay? Survive the harsh conditions for a long time. Essentially, endospore protects the genetic information of the microbe. You can see it right here. The DNA, additional copy of the DNA, gets encapsulated in a special structure that is called forespore. For like before spore. And the forespore develops more and more layers of protective structures, like cortex, for example, eventually forming a full several layers, the forespore layer, the cortex layer, and the protein coating on the outside. And essentially the endospore here, you can see, it contains only genetic information, practically no cytoplasm, no ribosomes. Think about endospore as a seed of a plant. Make sense? Endospores themselves do not replicate. Does that make sense what I just said? If you have an endospore, it's like a plant seed. It's going to be sitting there, not doing anything. Endospores by themselves do not replicate, they do not multiply, they just sit. But they can sit through dry, heat, radiation, cold, whatever. Does that make sense? Chemicals. They are very, very resilient. This process that's outlined on this scheme is called sporulation. So, formation of the endospore is sporulation. If endospore is formed in, say, dry environment, what would stimulate endospore to become back a normal vegetative cell? Moisture, exactly. So the process when it becomes a vegetative cell is called germination. Okay, in the process of germination, endospore becomes vegetative cell. Again, think about plants. You have a plant seed. You put it on the dry surface. Nothing happens to it. You put it in the soil, which is moist and has a lot of nutrients. And plant seed starts to grow, right? Does that make sense to you? So endospore requires some nutrients, organic molecules, and moist. And then it starts to germinate. Now if you put the vegetative population into the dry environment again, they will sporulate back. Now you can see the micrograph of the endospore itself. That's electronic transmission microscopy. And this is the endospore staining of some bacillus species. You can see the blue endospores in the cells. Okay? Why we talk about endospores so much? Because a lot of pathogenic microbes of two genera that we are especially interested in, the genus Bacillus and the genus Clostridium, form endospores. Bacillus anthracis causes anthrax. Clostridium, members of that genus, can cause tetanus, botulism, C. diff, Clostridium difficile, okay? So a lot of pathogens, Clostridium perfringens, the gas gangrene causative agent. A lot of pathogens are endospore formers. So we have to be aware of that. And endospores are extremely hard to kill. So we have to think about the control measures that we implement when we try to control for endospore presence. Does that make sense to you? Okay, so it's not just boiling. We have to autoclave stuff. We have to kill endospores in order to prevent the spread of those diseases.